Hello, my name is Brad Malpert, and this is my presentation on signal transduction and how it relates to the cell cycle regulation as part of the Concepts of Biochemistry course through Grand Canyon University and Dr. Hector Rivera. Today, we'll be talking about the cell cycle, which is the process cells go through to grow, prepare for division, and create new cells. The cell cycle is essential for development, tissue repair, and replacing old or damaged cells. Without it, our bodies could not grow or heal properly. The cell cycle has four major phases: G1, S, G2, and M. Together, G1, S, and G2 make up a stage called interphase, where the cell is not dividing but is actively preparing for division. The last stage of the cell cycle, M phase, stands for mitosis, which is when the cell actually splits. Let's break down interphase. In the G1 phase, the cell grows in size and makes new proteins and organelles. This is like the cell's building stage. Then comes the S phase, or synthesis phase, where the cell copies its DNA. Each chromosome is duplicated so that when the cell divides, both new cells will have complete genetic information. After that is the G2 phase, when a cell continues to grow and checks for any DNA errors that need repaired before dividing. Next is mitosis, the M phase. During mitosis, the cell organizes and separates the duplicated chromosomes into two identical sets. Mitosis has its own smaller steps: prophase, metaphase, anaphase, and telophase, followed closely by cytokinesis, which divides the cytoplasm. The result is two genetically identical daughter cells. The cell cycle also has built-in checkpoints that act like quality control systems. At the G1 checkpoint, the cell decides if it's ready to divide, checking things like size and nutrients. The G2 checkpoint makes sure DNA was copied correctly. And finally, the M checkpoint confirms that all chromosomes are properly attached to the spindle before separation. If problems are found, the cell can pause or fix them, or if the damage is too great, the cell may self-destruct through apoptosis. These checkpoints are critical for preventing uncontrolled growth, such as cancer. In summary, the cell cycle is a carefully regulated process that ensures cells grow, duplicate their DNA, and divide accurately. Its phases and checkpoints work together to keep our bodies functioning and healthy. Now that we've discussed the phases and checkpoints of the cell cycle, let's take a closer look at what actually controls the timing of those events. Two key players are cyclins and cyclin-dependent kinases, or CDKs. These molecules act like the engines and gears of the cell cycle, making sure everything happens in the correct order. Cyclin-dependent kinases, or CDKs, are special enzymes that add phosphate groups to other proteins, a process called phosphorylation. This phosphorylation changes how those proteins behave, essentially turning them on or off. However, CDKs are not always active. On their own, they stay inactive. They only become active when they bind to a partner protein called a cyclin. So, when are CDKs active? They're active only at specific times in the cell cycle, and that timing depends on the availability of cyclins. Cyclins are produced and destroyed in a repeating cycle, which is where they get their name. Their concentration in the cell rises and falls depending on the phase of the cycle. For example, G1 cyclins are present during early growth. S-phase cyclins appear when DNA needs to be replicated, and meiotic cyclins peak right before mitosis. This brings us to cyclins themselves. Unlike CDKs, which are always present in the cell at relatively constant levels, cyclins are tightly regulated. Their concentration increases when the cell needs to move into the next phase, then decreases after that task is complete. This rise and fall pattern is essential for keeping the cycle orderly. If cyclins were always around, CDKs could push the cell into division at the wrong time, which would be dangerous. So, how does cyclin binding affect CDKs? When a cyclin binds to a CDK, it changes the CDK shape so that it becomes active. This activated CDK cyclin complex can then phosphorylate target proteins needed for a specific stage of the cell cycle. For instance, cyclin CDK complexes in S phase activate proteins that start DNA replication, while meiotic cyclin CDKs activate proteins that control chromosome alignment and separation. In short, cyclins and CDKs work together like a molecular on and off switchboard. CDKs are always present, but require cyclins to activate them, while cyclins appear and disappear in carefully timed waves. This partnership ensures that each stage of the cell cycle happens in sequence and only when the cell is ready. If cyclin CDK regulation fails, the result can be uncontrolled cell growth, which is one of the hallmarks of cancer. To summarize, CDKs are the core drivers of the cell cycle, but they only work when paired with cyclins. Cyclin levels will rise and fall depending on the phase, and together with the CDKs, they control the precise order of the cell cycle events. Now that we've seen how cyclins and CDKs control the timing of the cell cycle, let's take a closer look at how their activity is fine-tuned. This regulation is mainly carried out by phosphorylation, kinases, phosphatases, and inhibitory proteins. First, phosphorylation is one of the main ways cells turn proteins on or off. It works by adding a small chemical group called phosphate to a protein, which can either activate or inactivate that protein's function. In the case of CDKs, phosphorylation acts like a brake or a gas pedal, depending on where the phosphate is attached. One important brake on CDK activity is Wee1 kinase. Wee1 adds an inhibitory phosphate to CDKs, which keeps them turned off until the cell is truly ready to move forward. This prevents the cell from entering mitosis too early. Think of Wee1 as a traffic light that stays red until the conditions are met. On the other hand, the CDC25 phosphatase does the opposite job. It removes the inhibitory phosphate from CDKs, which allows them to become active and push the cell cycle into the next stage of the cycle. You could imagine CDC25 as the green light that tells the cell it is safe to proceed. Another layer of control comes from CDK inhibitors, or CKIs. These are proteins that bind directly to cyclin CDK complexes and block their activity. Unlike Wee1 or CDC25, which use phosphorylation, CKIs act more like a physical barrier that prevents the CDK from working. CKIs are especially important in slowing down the cycle when DNA is damaged, giving the cell time to repair before dividing. Some well-known inhibiting proteins include P21, P27, and P16. The protein P21 is activated by the tumor suppressor gene P53 and blocks CDK activity when DNA damage is detected. This is a crucial safeguard against passing mutations onto new cells. P27 helps regulate normal cell growth by keeping cyclin CDK activity under control in early phases of the cycle. P16, meanwhile, acts mainly at the G1 checkpoint to stop cells from moving forward if conditions are not right. All of these controls, phosphorylation by Wee1, dephosphorylation by CDC25, and inhibition of CKIs like P21, P27, and P16, all work together to keep CDKs in check. This balance ensures that the cell cycle only moves forward when the timing is correct and the DNA is intact. If these regulatory systems fail, CDKs can become overactive, which may lead to uncontrolled cell division and cancer. To summarize, CDK activity is tightly regulated at multiple levels. Phosphorylation acts as an on-off switch. Wee1 and CDC25 set the pace, and CKIs provide emergency brakes. Together, they create a highly reliable system that protects the integrity of the cell cycle. Now, let's focus on one of the most important control points in the entire cell cycle, the G1S checkpoint. This checkpoint acts like a commitment point, where the cell decides whether it will enter S phase and begin replicating its DNA. Once the cell passes this checkpoint, it is essentially committed to completing the cycle, which makes this step tightly regulated. A key regulator here is the RB protein, which stands for retinoblastoma protein. RB is a tumor suppressor, meaning it prevents uncontrolled growth. Its main role is to control whether the cell moves from G1 into S phase. You can think of RB as a security guard at the gate between G1 and S. When RB is in its hypophosphorylated state, meaning it has very few phosphate groups attached, it is active. In this state, RB binds to a transcription factor called EF2. EF2 normally turns on genes required for DNA replication, but when RB is bound to it, EF2 stays inactive. This means hypophosphorylated RB blocks the cell from starting S phase. In other words, RB holds the cell back until the right signals are present. For the cell to move forward, RB must be inactivated. This happens through phosphorylation carried out by cyclin CDK complexes. First, cyclin D/CDK4 begins to phosphorylate RB during early G1. This partially weakens RB's grip on EF2. Later, cyclin E/CDK2 adds more phosphate groups, which fully inactivates RB. Once RB is fully phosphorylated, it can no longer inhibit EF2. With RB released, EF2 is free to activate genes needed for DNA synthesis, allowing the cell to transition into S phase. This carefully staged process ensures that DNA replication begins only when the cell has grown enough and external growth signals are present. The importance of this system is clear when it fails. If RB is mutated or missing, EF2 becomes constantly active, pushing the cell into uncontrolled replication. This loss of control is strongly linked to the development of cancer, especially retinoblastoma, a childhood eye cancer where RB was first discovered. In summary, the G1S checkpoint is a critical control point. Hypophosphorylated RB keeps E2F inactive, preventing premature DNA replication. Cyclin D/CDK4 and cyclin E/CDK2 phosphorylate RB step by step, inactivating it and releasing EF2 to start the S phase. This system balances growth signals with strict control, ensuring cells only divide when conditions are right. After a cell has passed the G1S checkpoint and finishes DNA replication in S phase, it faces another critical decision point, the G2 checkpoint. This checkpoint makes sure that DNA has been completely and accurately replicated before the cell enters mitosis. If there are mistakes or damage in the DNA, the G2 checkpoint stops the cycle so repair can occur. Without this safeguard, cells might divide with broken or incomplete DNA, which could lead to mutations or cancer. A major player in driving the cell past this checkpoint is the complex of cyclin B bound to CDK1. CDK1 is a kinase that, when paired with cyclin B, forms what's called the maturation promoting factor, or MPF. This complex acts like the ignition switch for mitosis. When cyclin B CDK1 is activated, it triggers processes such as chromosome condensation, breakdown of the nuclear envelope, and formation of the meiotic spindle. Importantly, cyclin B levels rise during G2 and peak at the start of mitosis, ensuring the timing is tightly controlled. Once the cell enters mitosis, another control system kicks in, the M checkpoint, also known as the spindle checkpoint. This checkpoint ensures that every chromosome is correctly attached to the meiotic spindle before the cell moves into anaphase. The spindle fibers must attach properly to each chromosome's centromere so that when they are pulled apart, each daughter cell receives a complete set of genetic material. The spindle checkpoint works like a surveillance system. If even one chromosome is not attached correctly, the checkpoint halts the cycle by blocking the activation of the anaphase promoting complex, or APC. This pause gives the cell time to fix the problem. Only when all chromosomes are aligned and attached does the APC activate, allowing sister chromatids to separate and the cell to continue division. Together, the G2 checkpoint, cyclin B CDK1 activation, and the M spindle checkpoint act as powerful quality control steps. They prevent cells from dividing with damaged DNA or unequal chromosome distribution. When these systems fail, the risk of genetic instability and cancer increases dramatically. We've talked about checkpoints and cyclins, but another important part of the cell cycle control is how the cell destroys proteins at just the right time. This process is handled by large protein machines called ubiquitin ligases, which take specific proteins for destruction. Two of the most important are the SCF complex and the anaphase promoting complex or cyclosome, or APC/C. The SCF complex works earlier in the cycle, especially at the G1S transition. Its main job is to target CDK inhibitors, or CKIs, for degradation. By marking CKIs with ubiquitin, SCF sends them to the protozoum where they are broken down. Once CKIs are removed, cyclin CDK complexes are free to drive the cell forward into S phase. In this way, SCF acts like a clean-up crew that clears away the brakes so the cell can move ahead. Later in the cycle, during mitosis, the cell relies on the APC-C. This is another ubiquitin ligase that plays a key role in the metaphase to anaphase transition. APC-C marks certain proteins for degradation, and by doing this, it helps trigger chromosome separation. Two important targets of APC-C are cyclin B and SERCIN. Cyclin B normally activates CDK1 to drive mitosis, but to exit mitosis, cyclin B must be destroyed. APC-C takes cyclin B for degradation, which inactivates CDK1 and allows the cell to finish division. Securin, on the other hand, keeps the enzyme separase turned off. Separase is needed to cut the protein glue that holds sister chromatids together. When APC-C destroys securin, separase is released, and the chromatids can separate. This carefully timed destruction ensures that chromosomes are evenly divided between the two daughter cells. In summary, both SCF and APC-C are like the demolition crews of the cell cycle. SCF removes inhibitors to promote the start of the S phase, while APC-C removes cyclin B and securin to allow the cell to separate chromosomes and exit mitosis. These systems highlight how the cell cycle control depends not only on building up proteins like the cyclins, but also on breaking them down at exactly the right time. One of the most important proteins in protecting the cell is p53, which is often called the guardian of the genome. Its main role is to monitor the health of DNA and make sure cells don't divide when their DNA is damaged. If p53 didn't do its job, cells could pass mutations to the daughter cells, leading to cancer. So, how is p53 activated? Under normal conditions, p53 levels in the cell are low because it is constantly degraded. But when the cell experiences stress, Like DNA damage from radiation or chemicals, checkpoint pathways stabilize p53 and increase its concentration. This activation allows p53 to act as a transcription factor, turning on specific target genes. The effects of p53 depend on how severe the damage is. If the damage is mild and repairable, p53 activates genes that pause the cell cycle, giving the cell time to fix its DNA. But if the damage is too severe, p53 can push the cell toward programmed cell death, or also known as apoptosis, to help prevent the spread of harmful mutations. One of the most important genes p53 activates is p21. The p21 protein is a CDK inhibitor, also known as a CKI, that binds to cyclin CDK complexes and blocks their activity. By doing this, p21 prevents the cell from moving past the G1S checkpoint. This pause gives the DNA repair machinery time to correct errors before replication begins. If repair is not possible, p53 triggers apoptosis, or controlled cell suicide. During apoptosis, the cell breaks down its contents in an orderly way and is removed without harming neighboring cells. While it may sound destructive, this process is essential for protecting the organism as a whole. By removing cells with damaged DNA, p53 prevents mutations from being passed on, reducing the risk of cancer. Now let's talk about the cell cycle is directly linked to cancer. Cancer develops when normal control systems in the cell cycle break down, leading to uncontrolled growth and division. This happens through cell dysregulation, where the normal checks and balances of the cell cycle are lost. A major cause of dysregulation is mutations in key regulatory genes. Normally, the cell cycle has strict checkpoints, cyclin CDK controls, and tumor suppressors that prevent errors. But when these genes mutate, the result is uncontrolled proliferation, meaning cells divide without stopping. Instead of pausing to repair DNA or waiting for growth signals, mutated cells push forward, which is one of the hallmarks of cancer. One important group of genes involved in this process is proto-oncogenes. In healthy cells, proto-oncogenes promote normal growth and division. But when mutated, they become oncogenes, which push the cell to divide constantly, even without proper signals. For example, mutations in growth factor receptors or signaling pathways can drive unchecked cell cycle progression. Mutations can also occur in the proteins that directly regulate the cycle, like CDKs. Normally, CDKs only become active when paired with cyclins, and when checkpoints give the all clear. But mutated CDKs can remain active at the wrong times, pushing cells into phases of the cycle prematurely. This adds to the risk of errors being passed on. On the flip side, cancer can also result from mutations in tumor suppressor genes, which normally act as brakes. Two of the most famous examples are p53 and RB. p53 is the guardian of the genome, as we discussed earlier. However, when DNA damage occurs, p53 halts the cycle or triggers apoptosis. A mutated or missing p53 means cells don't stop for repairs and continue to divide with this damaged DNA. RB, or retinoblastoma protein, normally blocks the transcription factor E2F to prevent entry into S phase. If RB is mutated, it can't restrain E2F, and cells rush into DNA replication without control. Together, mutations in proto-oncogenes, CDKs, and tumor suppressors like p53 and RB disrupt the delicate balance of cell cycle regulation. This leads to the uncontrolled cell growth that defines cancer. To summarize, cancer is not caused by one mistake, but by the accumulation of mutations that tip the scale, turning on growth-promoting pathways while turning off safety brakes. This is why studying the cell cycle is so important for understanding and treating cancer. Let's take a quick moment and review the big picture of the cell cycle and why it is so important. The cell cycle is the process by which cells grow, replicate their DNA, and divide into two daughter cells. It is divided into phases G1, S, G2, and M, and is tightly controlled by proteins like cyclins and CDKs to ensure everything happens in the correct order. Critical control points known as checkpoints are like the traffic lights. They pause the cycle if DNA is damaged, if replication is incomplete, or if chromosomes are not aligned correctly. The G1S checkpoint prevents faulty DNA from being copied. The G2 checkpoint ensures DNA is fully replicated before mitosis, and the M checkpoint checks spindle attachment during division. Proteins play a central role in this regulation. Cyclins activate CDKs, which drive the cycle forward, while inhibitory proteins like P21 and P27 can slow it down when needed. One of the most critical regulators is P53, often called the guardian of the genome. When DNA damage occurs, P53 can activate P21 to stop the cycle or trigger apoptosis if the damage cannot be repaired. When these systems fail, the result can be cancer. Mutations in proto-oncogenes, CDKs, or tumor suppressors like that P53 and RB remove the balance between growth signals and safety brakes. This leads to uncontrolled proliferation, which is the hallmark of cancer development. In summary, the cell cycle is normally a precise and protective process, but when its checkpoints and regulators break down, cells lose control and cancer can arise. This is why understanding proteins, checkpoints, and the role of P53 is essential for both cell biology and medical research. To help reinforce and visualize some of these concepts we've covered, here are two videos that can help further your learning. The Cell Cycle and Cancer by the Amoeba Sisters, and then The Cell Cycle and Its Regulation by Professor Dave Explains. Both these videos provide an engaging and clear overview of the cell cycle, its checkpoints, and how dysregulation can lead to cancer. And here are the references I used to make this presentation.